If one were to ask how could the DNA and the cellular mechanisms of simple cells, such as simple bacteria or even pre-cells, become those of the most complex cells alive today, including human cells, this certainly seems like an impossible task. However, when we study genes and genetic systems, we see so many of the processes which could have led to this development. So, for example, genes are duplicated, producing gene families. This is one of the major discoveries to come out of genome studies. Simply that our tens of thousands of genes are not individual representations isolated from all others, but rather genes get duplicated again and again to form gene families. Also, because genes typically come in pieces, separated by what are called introns, these coding sequences can be shuffled in a variety of ways to produce a large number of combinations of genetic messages from a small number of pieces. Also, the functional pieces of genes and proteins, known as domains, these are often shuffled so that one domain can be added to a second domain to create a new function. Obviously, this gets into more complex themes of genetics and molecular biology. However, our understanding and our study of molecular biology has led to an understanding of the processes which could convert very simple genetic and cellular systems into much more complex ones. And as we can compare genetics and molecular systems across the groups of living things, this is in fact what seems to have occurred through evolution.